Excuse me. Biomechanics of skeletal muscle is the study of how skeletal muscles generate force and produce movement by contracting and pulling on bones, essentially acting as levers to create motion. It's controlled by the nervous system and achieves coordinated movement throughout the body. It involves the understanding of the interaction between muscle fibers, tendons, joints, and the forces generated during muscle contractions, taking into account factors like muscle length, velocity, and the arrangement of muscle fibers within the muscles itself. The characteristics of muscle tissue are these four: contractibility, extensibility, elasticity, and excitability. Together, this means a muscle can respond to a stimulus, contract and shorten, be stretched, and return to its original length after stretching. So, contractibility is the ability of a muscle to shorten and generate force when stimulated. Extensibility is the ability of a muscle to be stretched without damage. Elasticity is the ability of a muscle to return to its original length after being stretched. And excitability is the ability of a muscle to the ability of a muscle to respond to a signal from the nervous system. Skeletal muscle is a type of muscle which has multiple nuclei within a single cell and is under conscious voluntary control, allowing deliberate movement of the body. So, for example, if you decide to lift your arm, the skeletal muscle fibers in your upper arm contract voluntarily, allowing for the movement due to the nucleated structure. It is also striated. This is very much unlike smooth or cardiac muscle, which functions involuntarily. All right, so skeletal muscles, like I just said, are voluntary. They're consciously controlled by the nervous system. They are made up of fascicles. These fascicles are bundles of muscle fibers which are surrounded by tissue, connective tissue, blood vessels, and nerves. So here's the muscle fiber, and then here's the fascicle. Okay, so this is one that are a bundle inside. Here's the muscle. This is the fascicle that makes up the muscle, and then inside of that is the muscle fiber. A single muscle fiber, here you go. A single muscle cell is called a muscle fiber. The membrane surrounding the muscle fiber is the sarcolemma, so you can see that here. And the fluid inside the muscle cell is called the sarcoplasm. The sarcoplasm is what contains the nuclei and mitochondria along with the myofibrils. The myofibrils contain two types of protein filaments that are arranged in a striated pattern, and that striation is what the skeletal muscle is named for. So here's your myofibrils, okay? All right, this is a little bit better of a picture of it. So here's your muscle fiber, and then it's pulling one microfibril out of it. So the sarcomere is here between these two Z lines. Each sarcomere is made up of thin and thick filaments, which are strands of protein, and they are called myosin and actin. So you can see the thick filament is the myosin, and the thin filament is the actin. Here is a better picture. All right, so here's the whole sarcomere. The thin is the actin, the thick is the myosin. All right. There are, I'm just going to pop down to this one quick too. Thick myosin, thin actin. Then there are protruding heads on the myosin filaments. These that form cross bridges, and they bind to the actin filaments and ATP, which is an energy transport molecule. These thin filaments here, the actin, have myosin binding sites. All right, so I'm going to flip back to this one for a sec. There are visible bands and lines in the skeletal muscle that are visible on with a microscope. The sarcomere is compartmentalized between two Z lines. So here's a Z line, here's a Z line. The sarcomere is the basic structural unit of the muscle fiber. Each sarcomere is bisected by an M line, right? And then the A bands here, that whole thing, contains the thick, rough myosin filaments, and each one is surrounded by 6 thin, smooth actin filaments. And then the I band contains only actin filaments. In both bands, the protein filaments are held in place by attachment to Z lines, which adhere to the sarcolemma. In the center of the A band are the H zones, which contain only myosin filaments. All right, so you can see that again here. Okay, so the sarcomere is here. It's compartmentalized between the two Z lines. Each sarcomere is bisected by an M line. Okay, and the A band, which is here, contains the myosin filaments surrounded by the actin filaments, and then the I band, again here, contain only the actin filaments. And then in the center of the A band is the H zone, and that H zone contains only the myosin filaments. Okay? All right, let's go back to this. This is just another picture of the myosin with the protruding heads, and those are what attach to the thin filaments, on the actin. This is just a bigger picture. Here's the myosin, the protruding heads, it's a picture of that. And then here is the actin that have the binding sites for those protruding heads. And then a microscopic picture of it. Myosin, the protruding heads, okay, and then the actin, and that's where they form those cross bridges. All right, so this is a picture of a fully relaxed sarcomere of a muscle fiber, and then a fully contracted one. All right. Excuse me. During muscle contraction, the thin actin filaments from either end of the sarcomere slide toward each other. The Z lines move toward the A bands. Those A bands maintain their original size. The I bands narrow and the H zone disappears. This is where the projections from the myosin filaments, which are called cross bridges, form the physical links with the actin filaments during the muscle contraction. All right, so you can see it's spread apart here. It's all put together here. Okay. All right, so there are several layers of connective tissue that provide the superstructure for the muscle fiber. The epimysium is a layer of connective tissue that surrounds the entire muscle. It protects the muscle, it's right here, protects the muscle from friction with other muscles and bones, and it helps the muscle to contract and move. Okay. The sliding filament theory is an explanation of how muscles generate force and movement through the interaction of actin and myosin filaments. Basically, in very simple terms, after a nerve impulse enters a muscle, the thin actin and the thick myosin filaments slide together within the muscle fiber, causing the muscle to contract or shorten. The next two slides are a description of the steps of muscle contraction, and then there's actually a schematic of it. Okay, so let's kind of break it down. So a motor nerve fires an impulse or an action potential. It travels to the neuromuscular junction, depolarizes the sarcolemma, and generates an action potential that spreads through the sarcolemma into the T-tubules, which excites or initiates the muscle fiber. The action potential travels down or propagates the sarcoplasmic reticulum to the sarcomere. The action potential causes release of calcium ions from that sarcoplasmic reticulum, which then diffuses into the sarcomere. The calcium binds to the troponin, which exposes active sites of the actin. Then those cross bridges are formed between the myofilaments. So again, between the myosin and the actin. Stored energy is released. That acts as the fuel for the power stroke. That's what the sliding action of the actin over the myosin. This is what shortens or contracts the muscle. New ATP binds to the myosin. It releases cross-bridging, releasing cross-bridge, returning to its normal position. The ATP breaks down into stored energy. If calcium is still present, the myosin reattaches to an adjacent region of the actin. So think about the myosin heads are like walking down the actin, pulling it closer together. Steps 5 through 8 continue and cycle as long as calcium is still present. Eventually, the sarcoplasmic reticulum reclaims the calcium, closing the active sites of actin, therefore relaxing the muscle. So this is a picture of that cycle. So you got up here excitation. So it's where a motor nerve fires an impulse or an action potential. It travels to the neuromuscular junction. That's where the depolarization of the sarcolemma happens, which generates an action potential that spreads to the sarcolemma. It's the, let me say that again. Once the action potential travels to the neuromuscular junction, it depolarizes the sarcolemma and generates an action potential that spreads through the sarcolemma into the T-tubule and excites the muscle fiber. Then the action potential travels to the sarcoplasmic reticulum, and the action potential causes release of calcium ions into the sarcomere. The calcium binds to the troponin and exposes the active sites of actin. That's where the cross bridges are formed. The released energy causes the myosin heads to rotate and pull the thin filaments inward, sliding past the thick filaments and shortening the sarcomere. The stored energy is released and acts as the fuel for the power stroke. Again, that's what shortens or contracts the muscle. Okay. New ATP binds to myosin, releasing cross bridge, returning it to a normal position. ATP breaks down into stored energy. If calcium is still present, the myosin reattaches to an adjacent region of the actin. That's that walking down. The myosin heads are walking down the actin, pulling it closer together. These here, 5 through 8, continue and cycle as long as calcium is present. And then eventually, the sarcoplasmic reticulum reclaims the calcium, closing the active sites of actin, therefore relaxing the muscle. Okay, so that's the whole steps of muscle contraction. All right. There are 3 types of skeletal muscle fibers. Type 1, which is slow twitch oxidative. Type 2A, fast twitch oxidative. And type 2B, fast twitch glycolytic. Type 1 muscle fibers, or slow twitch oxidative fibers, they contract slowly and primarily use aerobic metabolism, or oxygen, to produce energy, making it highly resistant to fatigue. Okay, so they contract slowly, rely heavily on oxygen to produce energy. And then due to this reliance on oxygen, they can sustain contractions for extended time without tiring easily. So for example, the soleus muscle during walking, or the back muscles for maintaining posture. Type 2A, or fast twitch oxidative, are fibers that contract quickly and use aerobic metabolism to generate energy, but may switch to anaerobic anaerobic, making them relatively resistant to fatigue compared to other fast twitch fibers, but they still fatigue faster than slow twitch fibers. So, for example, shoulder muscles used for throwing, leg muscles used for jumping. They're prevalent in sports like football, basketball, or tennis, where quick bursts of activity are needed with recovery between bursts. Type 2B, or fast twitch glycolytic fibers, they are also known as white fibers. You might see them written like that. They are used for short, powerful bursts of energy, and they produce the most intense contractions. They are very strong and fatigue quickly because they don't use oxygen, and they have a very large diameter. So, for example, muscles used in weightlifting or box jumps or sprinting. Type 2 fibers produce the most force. Okay? Almost every muscle in the body has a mixture of type 1 and type 2 muscle fibers, but the proportion of each type may vary depending on the muscle's function, the training adaptations, and the individual's genetics. The ratio of type 1 to type 2 fibers in a muscle is determined by its primary function. For example, muscles used for endurance activity like long distance running tend to have a higher proportion of type 1 fibers, while muscles used for explosive movements like sprinting have more type 2 fibers. Okay? Muscle fibers are organized into functional groups of different sizes. A motor unit is a single motor neuron and all fibers it innervates, acting as the functional unit of a skeletal muscle. When a motor neuron fires an electrical signal, all the muscle fibers within its motor unit contract simultaneously, producing muscle movement. The nervous system regulates muscle force by activating different combinations of motor units within a muscle. The number of fibers in a motor unit varies depending on the muscle's functions. Small motor units control precise movements like the eye muscles, and large motor units are for more powerful movements like what the leg muscles can do. Motor unit recruitment depends on the force or the resistance of the exercise. They are generally recruited in order of smallest to largest as the contraction increases. So, with light intensity exercise, type 1 slow twitch are recruited. As the force or velocity demands of the activity increases, the percentage of muscle fibers recruited also increases. The activation of more motor neurons will result in more muscle fibers being activated and therefore a stronger muscle contraction. The stages of contraction, also known as the phases of contraction, are twitch, summation, and tetanus. A twitch, which you can see here, is a single contraction of a muscle fiber, while summation and tetanus involve multiple contractions. A twitch is the contraction and relaxation of a muscle fiber after a single stimulation. A single action potential from a motor neuron will produce a single contraction in muscle fibers of its motor unit. This is an isolated contraction. A single twitch doesn't produce any significant muscle activity in the body. A series of action potentials to the muscle fibers is necessary to produce a muscle contraction that can produce work. So summation occurs when a muscle fiber is stimulated before it has fully relaxed from a previous twitch. So you can see that right here. So it's starting to relax and then it's stimulated again. Okay? If the fibers are stimulated while a previous twitch is still occurring, the second twitch will be stronger. Okay, so here's your first twitch, it starts to relax. Here's your second twitch, and it's stronger. Summation can be achieved by increasing the frequency of stimulation or by recruiting more fibers. Again, summation can be achieved by increasing the frequency of stimulation or by recruiting more fibers. The individual contractions become stronger with each stimulation. Again, you can see that here. All right, and that's a picture of your twitch, your wave summation, and then your tetanus. Okay, this is incomplete tetanus, this is complete tetanus. Tetanus occurs when the frequency of stimulation is so high that the muscle contracts continuously without relaxing. Tetanus can be incomplete or complete. Incomplete is when the muscle contracts quickly with a short relaxation phase. If the frequency of the motor neuron signaling increases summation, and subsequent muscle tension in the motor unit continues to rise until it reaches a peak point. The tension at this point is about 3 to 4 times greater than the tension of a single twitch. This is incomplete tetanus. Complete tetanus is when the relaxation phase completely disappears. So, for example, an incomplete tetanus would be like a light grip on a steering wheel, or holding a pen with consistent pressure, maintaining steady posture while standing. And then a complete tetanus is lockjaw or a back spasm. Okay. This is a microscopic image of a muscle fiber. All right, you can see the epiphyseal, all the structures here. You don't necessarily have to know this image, just know what is in there. All right. This is a great comparison of slow and fast twitch fibers. So you want to look at the contraction speed here, right? Fatigue rate and diameter. In the slow twitch, the contraction speed and the fatigue rate are both slow, and it has a small diameter. In fast twitch, both 2A and 2B have fast contraction speeds, but 2A has an intermediate fatigue rate and 2B has a fast fatigue rate. And 2A has an intermediate or moderate size diameter, 2B has a large diameter. Okay, so again, here's your slow twitch. Very slow contraction speed. It doesn't tire very easily, and it's a smaller diameter muscle. Type 2A, it's a very fast contraction speed. It does fatigue, not as fast as type 2B, but faster than type 1, has a moderate size diameter. And then your 2B, very fast contraction speed, very fast fatigue rate, and a very large diameter. Okay? All right. And then this last slide is an image of all the fibers together. Okay. That was a very quick, hopefully it was a review of some of what you learned in A and P. There will be a second video posted that Professor Diebendorf did on the rest of the PowerPoint and following along with the outline. As always, if you have any questions, please feel free to get in touch with me.